Good morning, everyone. My name is Prakash Puri. Today I am going to present my presentations on topic biological control of fall armyworm with entomopathogenic nematodes. My supervisor are Dr. Bree Wilson and Dr. Jessica. Without any delay, let's start the presentations. So, this is the outline of my presentation slide. So, starting from the research problem, fall armyworm is a highly destructive agriculture pest. So, it is rapidly spread from west to east and it was first identified in Western Africa. So, the 90% of the crop damage was observed in Queensland, NSW, and Western Australia inside the Australia. So, around 14.2 to 39.3 million Australian dollar damage per year was recorded through the fall armyworm. So within the 10 year of the outbreak, so it have a major damage recorded all around the globe. So the traditional control of the fall armyworms are such as the cultural control, chemical control, and integrated pest management technique. So the chemical control is the effective method, but it have a several downsides. So it have a resistance development, environmental and health risks, and the soil and water contaminations, and non-target species or organisms effect. So the alternative way to control the fall armyworms are entomopathogenic nematodes. So entomopathogenic nematodes are biological agents which have a potential for controlling insect pest populations. So it can kill the wide range of the insect species, and also it affect the highly effective against the larva of Lepidoptera insect order, and it was safe to the environment and safe to the consumers, safe to the farmers. So next, aims and objective. The aims of the study is to identify the effectiveness of entomopathogenic nematodes in controlling fall armyworm by examining the mortality rate of larva and pupa in laboratory and glasshouse condition. And the objective of the study is to identify the optimal concentrations of Sternemia carpocapsis, infective juveniles for effective control, and another one, the compare the effectiveness of entomopathogenic nematode in laboratory versus glasshouse environment to inform the field applications rate. So next, research questions. So there are three research questions we have been identified. So how effective are the entomopathogenic nematode species in controlling fall armyworm in Queensland? And another one is, what is the most effective dose of the commercially available entomopathogenic products for high fall armyworm mortality in laboratory and glasshouse condition? So last one, which fall armyworm life stage is most affected by the entomopathogenic nematodes? Are they different in effectiveness across the larva instar, or are pupa the optimal life stage for the entomopathogenic targeting? Background informations. Background informations of fall armyworm is a polyphagous pest. So it is reported in several countries. So it is one of the most serious pests in the maize productions. So it was first recorded in Australia in 2020. Due to its short life cycles and the long distance migration ability, they can spread very easily. So they can migrate like more than 100 kilometers per night. So the life cycle of the fall armyworms are the egg, larva, pupa, and the adults. So they can complete their life cycle in 23 to 27 days under the favorable conditions. So the larva stage is one of the most devastating or damaging stage of the fall armyworm. So they mostly are active during the dawn and the dusk time. The damage of the first and second instar larva looks like a transparent scratches and irregular winding on the leaves. As the larva develop or it enter inside the hole and start the feeding between the stem, leaves, and tassels. So faces can be clearly seen outside the whole of the plant, and it results in unmarketable cone combs and cause the high yield loss. So nematode species that can kill the insect are known as the entomopathogenic nematodes. So entomopathogenic nematodes have been shown to be very effective to control the several species of insect. So they can kill the insect in 24 to 48 hours, which is much faster as compared to the other biological methods. So the active stage of the entomopathogenic is third stage, which is also known as the infective juvenile. So this stage infect the host. So there are different families, like 23 different families of nematode that had a parasitic relationship with the insect. The species S. indica and the S. glacieri can infect and reproduce and complete their life cycle in 17 species of Lepidoptera, Coleoptera, and Hemiptera insect group. So also the several abiotic factors affect the entomopathogenic nematodes survival. Soil, sandy soil are the best soil for the survival of entomopathogenic nematodes. pH range 4 to 8 pH range is the average pH range for the entomopathogenic nematodes, and soil temperature, low soil temperature affect the survival rate of the entomopathogenic nematodes. Also, the study shows that the S. indica and the S. carpocapsi and the S. longicadum were highly effective against the older larvae and pupal stage as compared to the other species. So next, research design method. So in research design method, the location of the research is inside the University of Southern Queensland, Tomba, and the first process is collection and rearing of a fall armyworm. A colony will be collected from the AG Biotech company, and the larva, pupa, adult, and egg will be reared in the laboratory in a controlled temperatures. The temperature is around the 27 degrees Celsius, and relative humidity will be maintained at 65 to 70%, and the 12 is to 12 light and dark photoperiod. So after adult female lay egg will be transferred to the tray containing the artificial diet. Then after collecting, separating, and maintaining the larva, providing them artificial diet, and the last they will be transferred to the egg laying cage, and then after where they can start laying egg. At larva rearing stage, we provide the sweet potato as a food source to the larva. Also, we can see in the picture that the larva with the sweet potatoes, and the larva with the artificial diet, and the adult egg rearing cage. So next, treatment. So nematode treatment. The treatment will be include the different doses of the entomopathogenic nematode species Sternerima carpocapsi. So it will be sourced from the EcoGrow company. The concentrations of like 50, 100, 150, and 200 infected juveniles per ml will be used, and the control with the sterilized water are used. The each treatment will contain the 5 larvae, and this treatment are replicated at 3 times, and the control randomized design will be used in this experiment. So the experimental setup with the larva: 90 mm petri dish with the filter paper will be used. Each dish will receive the 1 ml of entomopathogenic nematode suspension using a pipette. So food is added to the vessels such as piece of sweet potato or discs of maize leaf. So vessels are sealed and providing the ventilations. The mortality of fall armyworm will be recorded every 24 hours for 5 days after the inoculation. So dead larva will examine for the presence of entomopathogenic nematode infective juveniles using the microscopes, and the color and texture of the cadavers will also examine and recorded. So the experimental setup with pupa is quite similar with the larva. Just difference is food is not added to the pupa. Only the chemo white tissue will be added for the pupa to hide, and vessels are sealed and ventilated. The same process will be repeated in the pupa as well. Next, glasshouse experiment. Sterilized topsoil, approximately 8.3 kg per pot will be used. They are arranged in design CRD. A cone plant of 3 to 4 leaf stage will be transplanted in a pot, and the 14 days after transplantation, a pupa will be applied to each pot in the soil. So after 14 days of the pupa applications, the treatment will be applied in each of the pot. So the mortality of a pupa will be recorded daily for 5 days, and the dead pupa will be examined, and the color and texture of the dead pupa will be recorded. So data analysis. The data collected from the various observed parameter will be entered and organized for analyzing using Microsoft Excel. So statistical tools such as SPSS and the ANOVA will be used for the analysis. We employ a 95% of confidence interval to determine if there are significant difference between the treatment. Next, expected outcomes. So the expected outcome, the study evaluate the performance of all fall armyworm on alternative food source such as the sweet potato to understand their feeding behavior. Also, the study helps to determine the optimal concentration of infective juveniles and evaluate the effectiveness of applying entomopathogenic nematodes for controlling fall armyworm larvae and pupa. And this information could be used to inform the application rate for the field trials in the futures. So, study also compare the fall armyworm pupa mortality under the laboratory and glasshouse conditions, and the research also promote the entomopathogenic nematodes as a biological method to mitigate the environmental and health impact of a chemical insecticides. So next, conclusion. So in conclusion, fall armyworm is a major global agriculture pest. It causes significant crop damage. It is the devastating pest uh, present all around the world. So, entomopathogenic nematodes show promise as a biological control method for managing fall armyworm population. So, the research helps to determine the optimal concentration of infected juveniles to effectively control the fall armyworm larvae and pupa. So, study also test the EPN concentrations and compare the pupa mortality and examine the feeding behavior. And study helps to promote the environmental friendly pest management strategy and reduce the chemical pesticide use. So these are the references. Thank you so much.